Afternoon, class, and thank you for attending your lecture. This afternoon, we're going to tackle the first topic of your midterms for hematology, lesson 6, which is anisocytosis. So, when you say anisocytosis, class, we refer to the so called variations in RBC size. Now, the first type of anisocyte class, or the first variation in red cell size, is your so called normocyte. Now, normocyte class, this would refer to your normal RBC size. And as you all know, your RBC would appear like a discoid cell. And they are often called discocytes. Normal size of 6 to 8 micrometer in diameter. And they have enucleated. When you say enucleated, they don't have a nucleus. It has a 120-day lifespan and has a small size, similar to that of a small lymphocyte. Ayos background natin, Miss Padadora. Diyos ko po. Anyways, copy. Okay, let's continue. So this is the appearance class of your normocyte. Anong WBC to, class? Tingin niyo. What WBC is that? Yep, lymphocyte. They are similar. Yung normocyte nyo or yung normal RBC nyo, they have a similar size with your lymphocyte. Okay. Now, the second type of anisocyte class is your macrocyte. When you say macrocyte, they are a larger RBC, greater than 8 micrometer in diameter. MCV is greater than 100 femtoliters. Causes are impaired DNA synthesis, stress erythropoiesis, excess surface membrane. And they are related with pernicious anemia, aplastic anemia, and cirrhosis of the liver. Please copy. Okay na? Sige, let's continue. So, this is your macrocytes class. They are larger than normal RBC. Let's go to the third one, your microcytes. So, microcytes class are cells less than 6 micrometer in diameter. They are small and round cell that can be found that can be due to fragmentation. Cause can be the cause of creation of your microcyte could be due to the failure of hemoglobin synthesis. Disorders related are your IDA, thalassemia, anemia of chronic disease, lead poisoning, and sideroblastic anemia. Please copy. Sino mo? Okay, let's continue. So this is your microcytes class. They would have a greater than one-third central pallor. Mas malaki yung central pallor nila. Now let's talk about the variations in hemoglobin content. So I'm including this class kasi it's one of the key factors that can affect your anisocytes. So variation in hemoglobin content, these are usually caused by shape of the cell. So kapag ang shape of cell nyo, if you have a smaller cell, smaller cell size, obviously you would have lower hemoglobin content, meaning you would be hypochromic. If you have a larger cell size, larger hemoglobin content, hyperchromic. Then degenerative changes, abnormalities of cell formation, particularly hemoglobin synthesis. Excuse me. Key terms. You have your so-called normochromic RBC or normochromic cell. These are RBCs possessing the normal amount of hemoglobin. They possess a central pallor which is about one-third of its diameter. Then you have your hypochromic cell. This is RBC wherein the central light area of the cell is larger and paler than normal. So yan yung sinasabi kanina na yung larger yung central pallor niya. And the cell would appear paler. Now there are two types of hypochromic cell. You have what you call hypochromia in erythrocytes with normal size, meaning they would have less hemoglobin in a normal shape RBC. So let me repeat, there is a type of hypochromic cell wherein there would be hypochromia meaning low hemoglobin in a normal size or normal shape RBC. Meaning less ang hemoglobin doon sa normal RBC na yan. Then we have your hypochromia in large RBCs or hypochromia in macrocytes. Meaning class, normal yung amount ng hemoglobin, but due to the large size of the cell, it produces a hypochromic effect. So let me repeat, the second type of hypochromic cell is hypochromia in RBCs with large size or macrocytes. They tend to appear hypochromic class because they would have the same normal amount of hemoglobin, but due to the larger size or the large size of the cell, it produces a hypochromic effect. Now, hypochromic is usually found in IDA, hemorrhages, parasitism, and malignancy. Please copy. So, dito sa parasitism class, there are some parasites class that are capable of causing microcytic, hypochromic anemia. Naalala niyo pa kung anong parasite to? You still remember? Naturo ba sa para niya yan? Hindi. Hindi si Palsipara. Bulate siya, bulate. Oo, yung hookworm. Your hookworms would cause this. So yeah, please copy. Self-explanatory naman class yung iron deficiency anemia sa inyo, di ba? Kung bakit siya nagkakos ng hypochromia. Di ba your hemoglobin is made up of heme and globin. Heme is iron, specifically the ferrous iron. Then the globin is the protein. So if you have iron deficiency in your body, you would have no, or you won't have enough heme to create your hemoglobin, leading to hypochromia. Sige, one more minute. Okay, let's continue. Now, another one would be your hyperchromic cells. These are RBCs which have an increased hemoglobin content and wherein the central light area is smaller than normal or non-existent. An example of this class is your spirocytes. Now, ang spirocytes nyo class, spirocytes class are a type of microcyte. Now, they are hyperchromic because the hemoglobin content is normal but has a reduced surface volume ratio. Meaning class, sa spirocytes nyo, normal ang hemoglobin content but you have a small or decreased size, making your hemoglobin content appear hyperchromic. Let me repeat. Your spirocytes are a type of microcyte. Now, these are cells that are hypochromic class, that are hyperchromic because they would have a normal hemoglobin content but has a reduced cell size, making them appear hyperchromic. Now, hyperchromic is usually found in megaloblastic anemia and hereditary spirocytosis. Then we have the so-called anisochromic. Now, when you say anisochromic, presence of both hypochromic and normochromic cells in the same blood film. Now, anisochromia is usually found in sideroblastic anemia and iron deficiency anemia after iron therapy. So kapag sinabi mong anisochromic class, you both have the presence of hypochromic and normochromic cells in the same blood film, seen in sideroblastic anemia, iron deficiency anemia, and even after iron therapy. Please copy. Okay, let's continue. Now, another type of variation class this time naman is in the staining property. Di ba gumawa na kayo ng blood smear, ng peripheral blood smear sa lab? Have you tried staining it, class? Nag-stain na ba kayo? Ha? Nag-stain na kayo, di ba? What's the stain you use? Anong stain ang ginamit? What's the composition of your right stain? Sige, let's start first with the fixative. Ano fixative niya? Methanol. Ilang seconds sa methanol, class? How many seconds? One. One dip. Okay, sige. Dip pala ang ginamit niyo. Okay, fine. After the methanol, what's next? Anong stain? Eosin. Now, ang eosin nyo, class, or your eosin Y, stains what part of the cell? Anong components? And your cytoplasm, class, is what? Acidic or basic? Okay, acidic components. Yung mga acidic components. Now, after eosin, what's next? Methylene. Methylene blue. Gaano katagal yung eosin Y, class? 5 seconds. Okay. How about methylene blue? Gaano katagal? 5 seconds din. Okay. Iba kasi yung alam ko sa lab at sige ko yun ang turo sa inyo. Tapos, after methylene blue, what's next? Talaga, water. Okay. Sa amin kasi buffer. May buffer na siguro pwede rin ng distilled water. Okay. Ang distilled water kasi, class, pag ginamit niyo yan, pwedeng mag-lyse yung RBC. Anyways, kung yan ang turo. Buffer ang ginagamit namin. Now, after staining it, class, you'll see variations on the color of your cells, di ba? So, sige. So, the first one, yung variation in staining characteristic is yung polychromatophilia. Refers to the appearance of cells after staining. Then, you have your polychromasia. Refers to the appearance of young cells after staining. So, pag polychromatophilia, normal cells or adult cells after staining. Pag polychromasia, appearance of young cells after staining. So, what do you think, class, is the color of young cells after staining? Yung buffer na yun, class, PBS lang yun. Phosphate buffer saline lang yun. Baka meron naman dun sa laboratory. So, anong kulay? What do you think would be the color of the young cells after staining? No, dalawa lang naman yun, di ba? Pink, blue. May nakita ba kayong kulay green, yellow? May nakita ba kayo? Wala naman, di ba? So, it's either pink or blue lang yan. Kapag pink, yung RBC nyo, what does it mean? Mature, di ba? Oo. So, madalas kasi ang appearance ng young RBCs nyo, young cells nyo, it either appear blue or they would appear dark. And aside from that, ano pang meron sila? Aside from the color, what else would you look for if you want to know if it's a young cell? Young cells have what class? Okay, they have nucleus. Anong kulay ng nucleus nila? Dark blue or blue. So, yun siya. Then we have your diffuse basophilia, refers to the presence of RNA contents that are scattered throughout the cytoplasm, stained with the various shades of blue with tinges of pink. Cells are larger than normal and would correspond to reticulocytes. Now, this type of variations class are found, itong diffuse basophilia nyo, is found in reticulocytosis, hemolysis, acute blood loss, and pernicious anemia. Please copy. Okay, let's continue. Then another term class is your punctuate basophilia or the so-called basophilic stippling in RBC. This is a type of polychromasia class in which basophilic granules are found in the cytoplasm of the erythrocyte. The reason for this is due to vascular degeneration of the polychromatic substance of the cytoplasm. Stippling represents aggregated RNA. Class, ang basophilic stippling niya would occur if there is abnormal RBC maturation. If you still remember the rule, as the cell matures, RNA is removed. Now, due to this abnormal RBC maturation, some aggregates or some amount of RNA would remain in the cytoplasm, forming this so-called basophilic granules or basophilic stippling. So let me repeat, your basophilic stippling is formed due to abnormal RBC maturation. This is the representation of aggregated RNA. And if you still remember the rule, as the cell would mature, RNA is removed. Now, due to the abnormal RBC maturation, some RNA is retained in the cytoplasm, leading to the formation of this so-called basophilic granules. Now, basophilic stippling or your punctuate basophilia is found in toxic anemia, hemolytic anemia, leukemia, thalassemia or your globin chain deficiency, and congenital forms of anemia. So please come. Okay na? Let's continue. Now, your hypochromasia class is when your red cells would stain or would appear pale. Now, this can be caused by lowered hemoglobin concentration and abnormal thinness of your RBC. So again, hypochromasia is wherein your red cells would stain when stained would appear pale, caused by low hemoglobin concentration and abnormal thinness of the red cell, found in IDA, sideroblastic anemia, and thalassemia. Please copy. Okay, let's continue. Hyperchromasia class. Now, this is when your red cells are stained deeply blue due to the thickness of the cell. So, dito naman class overly colored blue yung cell niya. Found in macrocytosis, spirocytosis, and megaloblastic anemia. Here's a question. Why do you think in this type of cell they would appear blue? What's the reason, class? Bakit siya naging hyperchromatic? If you understood bakit pink, blue yung mga cell nyo kapag ini-stain nyo siya, what's the reason? Bakit less acidic? So you would have here class higher nuclear content in the cytoplasm, leading to a thick cell. And remember, ang nuclear contents nyo, di ba, they are always stained blue. Found in macrocytosis, spirocytosis, megaloblastic anemia. Please copy. Let's continue. Then we have your anisochromasia. A proportion only of RPCs are pale. They are found in IDA, responding to iron therapy, and sideroblastic anemia. Please come. So I think class naintindihan nyo naman yung difference ng variation in hemoglobin content, di ba? Kapag hemoglobin content, it would be hypochromatic. Kapag another variation naman sa staining property, meron siyang hypochromasia. There is the word Misha and Matic naman for hemoglobin content. Kasi yung mga hemoglobin contents nyo, they affect the staining properties. Please copy. Okay, let's continue. That ends it. So I will post the recording class so you could view it.